Okay, did everything go okay yesterday? Did all your stuff inoculated? The gelatin is going to take a while because for most of you, 20 degrees Celsius is below, fairly well below your optimum temperature. So we'll probably have to leave those in there for a week or more before you can really see results from those. The other ones we'll evaluate on Wednesday. When you take your drop tubes out of the incubator, don't shake them all up because you want to see if there's a pellicle or if there's fine turbidity, if there's a sediment. So take those out carefully and look at them. Okay? And then you do need a 24-hour to 48-hour culture of your unknown. So the ones you inoculated yesterday, after you look at them, you could then use those as your inoculum for your first staining tests. Okay. Anybody read ahead? You know why we need a 24 to 48-hour culture of your unknown? Gram stain is time sensitive. Okay, if you don't get outside of that 24 to 48-hour window, you start losing reliability for your gram stain. So anytime you want to do a gram stain, you're going to have to be aware of that. Okay? Get in the habit of looking ahead for a couple of days before the next lab to see if you need a culture of a particular age, or check your cultures and make sure that there's enough growth there for the experiment we're going to do. I'll try to remind you, but it's also your responsibility. So, any questions? No? So, what process is being illustrated up on this slide here? That is binary fission, okay? That is how microorganisms grow. How do your parents measure when you grow?
Door.
What? Door, yes, yeah. Little lines on the door. What's another way to measure growth of humans and other animals? Mass, okay? So what is a researcher going to do if they're working with a particular species and they want to monitor the growth of the organism they're working with? Let's just be boring and say it's E. coli. Are they going to look at the size of an individual cell and follow it over time and measure it on a door or put it on a scale? Obviously not. They're too small, right? So what strategy do we typically use to measure microbial growth? Do we measure individuals? We measure populations, okay? We look at microbial populations and see how they grow and change with respect to time. So one of the questions is, how do we do that? How do we go about measuring the growth of populations over time? How do we count the number of cells in a given population at a given time? Now, you're already starting to get a little bit of a sense of how you can do this qualitatively by inoculating something into a drop tube and putting it in the incubator. And what's the first thing you do when you pull it out? What do you look for? If you want to know if you're successful, this is growing, how do you evaluate that? By looking at the tube. See if it's clouded. Okay, check for turbidity. And then we superimposed a quantitative aspect on that by putting the tubes in the spectrophotometer so we can actually get a numerical reading of the value of turbidity. And we'll talk about that strategy in just a minute. What's the term that refers to how long it takes a population to double? Because when you divide by binary fission, you double, right? One cell forms two. That term that refers to how long it takes a population to double is generation time. Does that vary from species to species? Yep, that is a species characteristic. Remember I mentioned that for some cells that grow pretty fast, populations that double pretty fast, generation time is 10 to 20 minutes. I mentioned that mycobacterium, which causes leprosy and tuberculosis, has a very long generation time. It's not very contagious. It could be days for the generation time. So it is somewhat of a species-specific characteristic. Are there other factors that can influence generation time besides the type of microorganism? Yeah, environment. So there are a number of environmental factors, one of which you've looked at a little bit already, temperature. Another one we'll investigate later on is osmotic concentration. So we'll take a look a little bit later on at some of the factors that determine how fast something can grow. There are some practical and somewhat scary reasons for wanting to know how fast microbial populations grow. There was the example I gave in the lecture notes, and I don't know if anyone's read it or not, but the scenario said basically, how long does it take, or how many microorganisms will you have, if you have an egg salad sandwich, and there are a few salmonella cells in that, about 10, if you let it set out for 4 hours, and those bacterial cells are allowed to divide and grow. Anybody look at that number? So salmonella, 4 hours, start with 10 cells. Calculation tells you that after 4 hours you have about 41,000 cells. Okay? Quite a few. This is the formula that allows you to determine the rate of growth of populations over time. N sub f equals n sub I times 2 to the nth power. I suppose n sub f refers to. Final number, okay. That's what the 41 table is to be. What's n sub I? Initial number, that would be the 10, okay. 2, we're okay with, but what's the exponent? What's n? N is how many generations it will have gone through. And what do we need to know? We need to know time, and in this particular example, it's expressed in minutes. Okay, so 240 minutes. What else do we need to know? Oops. What's missing? What information is missing right now from this calculation? Generation time. How long it takes for one generation to be produced. And we're going to assume in this particular case a pretty average generation time of 20 minutes. So the number of generations is the total time divided by the generation time. So n is 12. 10 times 2 to the 12th power gives you this 41,000 approximately. If we let that go for 6 hours instead of 4 hours, what do you think it would be? Well, here's some possibilities I want you to consider. How about 67,000? How about 327,000? How about 262,000? How about 1 million? What do you think? What's your guess? Okay, it's going to be a big number, okay? Why are you not looking at this one real seriously? This is about 1.5 times this value, and that's about 1.5 times as much time. It's exponential growth, okay? Anything that grows by doubling is exponential growth. That's the correct number right there. Because you're up to 18 generations. Okay. 6 hours is 360 minutes. 360 divided by 20, so 2 to the 18th power gets you up to this 262,000.
But that's an ideal condition.
That's an ideal condition, yes. Okay. And That's one of the things we always have to consider when we're looking at generation times and rates of growth is, are the conditions ideal or are they not? So this gives you a good example of what exponential growth looks like. 1 cell to 2, 2 to 4, 4 to 8, etc. And this gives you the exponential value of growth and the number of generations. But this is what the growth curve looks like if you're looking at exponential growth. And this is the line that we're going to be most interested in right here. This is a plot of the number of cells over time. And then if we turn that into a logarithmic plot, it straightens it out to a straight line. But this particular growth curve right here is characteristic of exponential growth. If you plot the number of cells over time. So let's break apart a typical exponential growth curve a little bit and see what's happening. Okay? So we get this characteristic kind of almost bell-shaped curve right here. And when we break that curve down and look at it, we can see some interesting things that are happening to the cells within the population. So let's just make pretty much a bell-shaped curve. Okay? The first phase of growth is called the lag phase. Now, let's put this in the context of you're transferring your unknown from an old slant into a fresh slant or a fresh broth. What's happening to those cells? Well, the first phase, there's not much growth that's occurring. We call that the lag phase. And there's a couple of reasons for that. One is that initially the number of cells that are being produced by population doubling is not that great. Let's say theoretically you really only started out with 1 cell that you were transferring. Okay? So in 20 minutes you'd have 2. Another 20 minutes you'd have 4. And so it can be hard to measure the number of cells when the populations are so small. But there's something else that's going on here. This is really a period of adjustment by the cells. By the time those cultures have gotten old and you figured, oh my gosh, I better transfer them so they're still viable, those cells have started to deplete nutrients. Toxins have begun to be produced. Many of them have responded to this by slowing down their metabolism. That might involve turning off genes that aren't needed right now because, for example, the food resources aren't available. So why make enzymes to break down those food resources if they're not available? So when you put them in this fresh new growth media, it's going to take some time for the cell to gear up and to really start maxing out again in terms of its reproductive capability. The next phase is called the phase of positive acceleration. So now we're starting to take off growth a little bit. The key here is that all the cells in that particular population aren't doing the same thing at the same time. Some have made their adaptation. Others have not yet. So some are growing at their maximum rate, but others haven't quite hit the maximum yet. So it'd be like if you had a bunch of race cars all bunched up together. Some have zoomed off, some are still getting going, okay? Or a bunch of runners in a marathon, okay? But not everybody's doing the same thing at the same time. There can be slight genetic differences in the cells that cause them to respond differently. It might be something like their position in the culture, okay? And what they're exposed to in that particular environment. When we get this pretty much linear growth right now, that indicates a constant rate. And really now, at this time, the culture's synchronized. Pretty much all the cells are clicking along. They're all dividing at their maximum rate. In this particular situation, the environmental conditions are pretty much ideal. Plenty of nutrients, right temperature, not many toxins. Using an ecological term, Dr. O'Neill would love me for that, these cells have achieved, or these populations have achieved their biotic potential. Okay? They're growing literally as fast as they can. So for the salmonella bacteria we use, that would be about every 20 minutes the population would be done with. But do things maintain ideal conditions forever? No, we're working in a closed environment. Okay? It's so cloudy. So now we get into this phase of negative acceleration. Are things synchronized now? No. Some cells have maxed out already and aren't growing at their biotic potential any longer. Some still are. And then we get into this period of time called the stationary phase. Things pretty much aren't changing. So basically, for every cell that's being produced as a result of cell division, what's also happening in that culture? Cells are dying. Okay? So if you want to think of this in terms of birth rate and death rate is equal to death rate. So really here, we're in an equilibrium. The number of cells that are present once you reach this stationary phase is the maximum number of cells that the environment can sustain. That's the maximum population density that you can get for that particular species in those particular conditions. Again, an ecological term. At this particular point, the population would have reached its carrying capacity. Now populations start to drop. And we start to enter this fairly prolonged period that's called the death phase. And the death phase can be quite protracted, and sometimes it can take a long time for those last few cells in this population eventually end up dying. Because one of the things that does happen is that as cells die, they become potential food resources for the living. A little bit of microbial cannibalism going on, basically. Okay? But what eventually is going to make this population crash and completely die out? Pardon me? Toxins. Yep. I mean, so even if a cell's being born and then another one's consuming its resources when it dies, toxins will eventually limit the growth of this population and cause death to occur. These organisms that hang around sometimes for a long period of time are called persisters. And oftentimes, these particular organisms have some slight genetic variation from their brothers and sisters in the population. that give them some advantage that allows them to live a little bit longer and tolerate those conditions a little bit more effectively. So sometimes these persisters are kind of mutants. Okay? Practically speaking, this is why you should never discontinue antibiotic use before you've taken your full course of treatment. And you certainly shouldn't say, oh, I'm feeling better. I will save some antibiotics for the next time I'm not feeling so good, and then I won't have to go to the doctor. When do you think you probably have your greatest symptoms? Which of these phases? During this log phase. This is when those cells are most active. Many of the things that they produce that cause disease are toxins, and so they're cranking out these toxins. When are these cells most susceptible to inhibitors like antibiotics? During this log phase, okay? Their metabolic rate is the highest. They're transporting things into the cells most effectively. Okay? So, if you take antibiotics, that basically forces this population to do that prematurely. And so once it levels off and starts declining, you feel better, right? But are their cells still alive? Yeah. And what kind of cells are they most likely? Ones that already have some inherent resistance to the antibiotics. So by doing that, you're basically selecting for the antibiotic-resistant forms. And then if you get the same infection next time, you might even get a rebound from this infection. And you say, well, I guess I better take the rest of my antibiotics. What's going to happen? Most of these cells that have rebounded now will be the more resistant forms. So it's not just that drug companies want to sell their antibiotics. There really is a reason for you to take these particular antibiotics over their course to make sure that you knock out pretty much all of the pathogens, including the resistant forms. When do you suppose, if you have an organism that's an endospore-producing organism, that it would probably begin producing endospores? Yeah, it might even start in the stationary phase a little bit, but definitely when it starts getting into this death phase, that's when you start switching over and going, uh-oh, time to produce some endospores and try to wait out these adverse environmental conditions. Okay, practically speaking, if I'm going to make a growth curve, how do I know the number of cells in a population at a given time? How do I generate one of these growth curves? That involves knowing at time x that there are a certain number of cells. So how do I go about counting microorganisms? Because you can't make growth curves if you don't know how many cells there are. Well, there are a variety of techniques that allow you to count microorganisms. And one of the things I want you to concentrate on now as we go through some of these is, number one, how does the technique work? Number two, what are some advantages of this technique over other methods? There are always trade-offs. What are some disadvantages? And then, when would you choose to use one method over another method? So what are the potential applications? Well, the most straightforward way to count microorganisms in a population is to do an actual direct microscopic count. And to do that, you obviously need a microscope. And you need a little device. It's got several different names. Petrov-Hauser chamber. It's also called a hemocytometer. Some of you might have used that with Dr. Morton. McCombs when she does her yeast cell counts. But basically, it's a device where you have a slide with a little indentation in it. And typically, that indentation will occupy a certain fixed volume. Okay, so maybe like 100 microliters. What that allows you to do is, if I've got this big flask of cells, I can take out a portion or an aliquot, 100 microliters, put it in this particular special slide, count the number of cells in that aliquot, and then multiply how many aliquots there are in the total volume. Okay? So I'm doing a sampling of the total. Now, when you do samplings like that, what assumptions do you make? That there's complete uniformly distributed cells within that solution, and that the sample that you take is representative of the whole, right? So you might want to vortex them, for example, as opposed to having a situation where you've got sediment in the bottom, and you take your sample from the top. These particular Petroff-Hauser chambers have little grids inscribed into the cover slip that covers up the little well. What's the purpose of having these little grids right here?
So that when they move around,
Yeah, it's really just a way to keep track of what you've counted and what portion of the slide you've counted. Okay. I don't know if you've noticed, remember, have you used the colony counter up there before? I thought we did that in cell one time where we counted the number of colonies. So there's a little grid on there, and so you can systematically go through the plate, grid by grid by grid, and count the number of cells, and that way you don't lose track as easily. Now, Zach brought up an interesting limitation of this particular technique. What is the limitation of this technique? If your cells are mobile, okay? So you count this guy up here, and then all of a sudden it moves down here again. And if they're all moving around, that's a problem. If the cells aren't mobile, is it really easy to tell whether these particular cells are alive or dead? Say it's just a little coxsite that's just sitting there. No. Now there are some stains you can do called vital stains, which have some ability to distinguish cells that are metabolically active versus those that are inactive. But maybe they just have a low state of metabolism. Maybe they're kind of in suspended animation. And so this particular technique has difficulty distinguishing living cells from dead cells. And that can be an issue. That can be important to know sometimes. Not just how many particles there are, but how many actual live cells there are. If it's a pathogen, for example. One of the things we're always interested in when we talk about some of these techniques and methods is expense. Do you think this is a very expensive proposition? No, I mean, you got a microscope, there's some expense to that. These actual little slides are not cheap, but you could have one of these in a doctor's office easily and count some simple populations by just direct microscopic observation. One of the other things that we're always interested in too is how much is a method prone to error? What do you think? Error prone or not so much? Yeah, I mean, just setting aside the issue of motility, this is a tedious type of task. So you're counting line by line by line. And if something is tedious, then it's more likely you're going to make mistakes. Okay? So pretty simple method, but prone to error and doesn't distinguish living from dead. Well, there's an electronic version of this basically called the Coulter counter. And as soon as I say electronic, what about cost? Way up, okay? So machines that try to automate things probably will increase reliability, right? But are going to be more expensive. And some machines are relatively difficult to operate, fairly sophisticated, and so you need more training, which also adds to the expense. The concept of the Coulter counter is really a pretty simple concept. You take a sample in liquid, so this would be an aliquot of a solution you were measuring for microbial counts. You put it in some kind of little orifice, and the cells are pulled through that little orifice, and there's an electronic detector coupled with this. And so every time a particle passes that detector, it breaks the light beam and registers a count. Okay? It's kind of like stepping on the automatic door, and the door opens for you. Okay, you're breaking a light beam. That triggers an event, in this particular case, a count. So, greater expense, probably less air. Is this going to distinguish living from dead? No, pretty much particle to particle. What if I were counting populations of E. coli versus populations of staph? Which one do you think I might get more reliable results from? Remember what E. coli are? They tend to be little single gram-negative rods. What's characteristic of staph, which are gram-positive cocci? They're round, and what does staph low mean? Remember, those are those clusters, those grape-like clusters, so the cells stick together. So one mass of several hundred staph cells passing through this orifice might be counted as one, right? So things that stick together could be underestimated by this particular technique. This is a faster counting method than direct microscopic examination. But this is a kind of tool you would typically find in a med tech lab as opposed to a doctor's office. Another technique that we can do that gives us some interesting information, particularly about live cells in a population, is the plate count. Now, theoretically, this is a pretty straightforward technique. So I've got some sample here. And I don't know number of cells. If I could determine number of cells and I know the volume, I could get a concentration, right? Cells per volume would give me a concentration. But I don't know number of cells. Is it necessary to measure this entire sample? No, you can take aliquots out of it, and assuming that there's a uniform distribution, I could then say, if this is a 100 mil sample, and I take out 1 mil and count the number in 1 mil, then I can just multiply that result by 100 to get my total sample size. Okay. So, what do I do in a standard plate count? Well, I have an agar plate. It's sterile. And I sterilely pipette in 1 milliliter, for example, of the sample. You could do any aliquot size you wanted to do. And put it on a plate and incubate it, and hopefully over time various colonies would grow. What does a colony come from? What produces a colony? Well, a colony is a bunch of bacterial cells clumped together, but what gave rise to that colony initially? Technically one cell, right? Isn't that what you're trying to do in your street plating? You are trying to isolate out one cell on the surface of that agar plate, and then put it in the incubator, and that cell grows, divides, exponential growth. And over a period of time, you then get a mass of cells. And what's the relationship of all those cells to one another? They all derive from the same cell. They're clones of that cell. Okay, so they should be genetically identical unless a mutation occurs. So, If I then count the number of colonies, and let's say I had 50 colonies, that would have represented 50 cells in that 1 mil aliquot. And then I could just multiply that by my factor of 100, and that would give me the total in the original population. What if I had too many? What if I had like 5,000 colonies? Would that be easy to count those? No, they'd probably be spilling over on top of one another. What if I only had 3? Then that's not many. You'd start worrying about statistical significance. So a plate is only considered to be countable if it has between 30 and 300 colonies. Now, if I only had 3, what would my strategy be if I wanted to get a countable plate? Put more on there, right? But at some point in time, I would probably exceed the ability of the plate to accept that volume. Okay? I couldn't fit all 100 mils on the surface of that one auger plate. So if I get too few colonies, one of the things you sometimes can do is just concentrate this. Okay? There's a device called a millipore filter. Have any of you worked with Chiquana and any of his water quality stuff? Groups of students that have worked on that have done bacterial counts of water, and they're fairly dilute. And so what you have to do is you have to take the original sample and basically pour it through a filter, and the sterile filter traps the bacteria, and then you put that filter on the plate. So basically you're counting all of the cells in the population. So if it's too few, you can do some kind of concentration method. What if you got too many? What if you got that 5,000? Then start doing smaller aliquots. Do 100 microliters or 10 microliters. But at some point in time, you run into limits of what you can accurately pipette. If you're going to try to take a 1 microliter sample out of this, it's tough to accurately pipette those small volumes. So typically what you do if you're counting large populations is you do a series of dilutions. You do serial dilutions. To do serial dilutions, you take a known aliquot, let's say 1 milliliter, and transfer it into a new tube of sterile broth. Sometimes you also use saline solution. If you do the saline solution, it's important that that saline be isotonic. Why would it be important for the saline to be isotonic? Yep, yeah, you can kill the cells if you didn't have the saline solution at an isotonic. And this particular technique is good because it's one of the ones that measures numbers of living cells in the population. Because if you're going to grow on that plate, the original cell that came out of the culture had to be viable. Okay?
So if 2 cells fell off on the street and 2 cells fell off right next to each other, they wouldn't start the same colony, they would still go to different colonies?
If they fell off right next to one another, they could overlap. And so the assumption is that an isolated colony is derived from a single cell, but you're right, there could be instances where they fell off and they were touching one another and they weren't modal, and therefore they could both contribute to the same colony growth. Yep, absolutely true. So let's say we took that 1 mil and put it into 9 mils of sterile broth or sterile saline. Okay. What is that dilution factor or step dilution factor for the first step? That's going to be a 1 to 10 dilution. You take, to calculate that, the amount of diluate, the stuff you're diluting, and divide it by the amount of diluate plus the amount of diluent you're using, in this case broth or same. So if you take 1 mil and put it into 9 mils, that's a 1 to the 10th dilution. I think it's much easier to do this exponentially and say it's a 10 to the minus 1 dilution. Maybe that would be sufficient, but in large populations, that might not be a sufficient dilution. So what can we do instead? We can then take an aliquot out of this tube and put it into another tube that also contains 9 mils. Okay? What's the step dilution going to be of this dilution? That's still a 1 to 10 dilution. So that's a 10 to the minus 1. But what is our total dilution now? It's the product of the two individual steps. And then we could do another 1 to 10. And our step would now be, or our total dilution would be 10 to the minus 3. You could then plate these each out on individual plates. And at some point, hopefully, you will get a countable plate. Let's say we got 50 colonies in this step dilution right here. Here's the formula then that you could use to calculate number of cells in the original sample if you got 50 colonies from this particular tube. CF is equal to CI sorry. I'm thinking well here. My concentration initial, CI, is equal to CF times DF. So my initial concentration is what I want to solve for. CF is basically the number of colonies. And you multiply that times 1 over the dilution factor. If the dilution factor is 10 to the minus third, 1 over the dilution factor is 10 to the third. So if I had 50 colonies, that would be 50 times 10 to the third cells. And you would probably simplify that and say, I've got 5 times 10 to the fourth cells. And because we knew we started out working in milliliters, that would be our concentration of the cells in milliliters. We're going to do this in lab. It's actually probably going to end up being an optional point activity. But what I'll give you is I'll give you a flask up here, and it will have an unknown concentration of cells. You will make a series of dilutions, and then for some of these step dilutions, you'll take a small aliquot out, put it on a plate. You'll grow it, count the number of colonies, and if it's in this countable range of 30 to 300, then you can use this formula to determine the number of cells. So this gives you a viable cell count. What are the disadvantages? Yep, possibility for contamination, possibility for not doing your step dilutions accurately. What's really going to determine kind of accuracy of this? What technique? pipetting, right? Okay. And if I'm doing a 100 microliters for my step dilution each time, and I'm only off by 10 microliters, that's a 10% error. So accuracy in doing your pipetting is really crucial to getting good results. Another problem with this is we've got to do this, and then we've got to incubate them overnight at least to get growth so we can do counting. And so this takes time. This is a slow method. The big advantage of this particular method is cost. It's really cheap to do. You can do auger plates and you have pipettes. And the ability to distinguish living organisms from dead organisms, you're in pretty good shape. Now, there are some other techniques as well that are much faster, pretty low tech, but they require the construction of a standard curve. And that's definitely the case of measuring populations by turbidity. So in which of these tubes are there more cells, left or right? Right, obviously, okay? There are cells in there, and those cells diffract the light and give that cloudy, turbid appearance. So you could say more cells here than here. Your eyes are fairly good at being able to do qualitative determinations. Well, what is the concentration of cells in this? No way to tell, right? You could use a spectrophotometer like we did and say this one has X times the number of cells that this does, because you get some quantitative numbers, but you still couldn't tell a concentration unless you had a standard curve to refer to. So what do we have to do? Well, using the spectrophotometer to measure the absorbance of these solutions is pretty straightforward. Well, what we have to be able to do is to determine the number of cells that give a particular absorbance under specific conditions. Again, think back to cell biology. I don't know if you remember this or not, but we had solutions of the purple potassium permanganate. And one of the things that I asked you to do was to take an unknown solution and give me its concentration. What did you have to do first? You had to take various solutions of known concentration, 4 milligrams per mil, 8 milligrams per mil, 16, and you had to do absorbance readings of all of those. And then you had to make a standard curve that plotted out the relationship between milligrams and absorbance. And then once you had that standard curve, you could take the absorbance reading of the unknown, plug it into the standard curve, and find out what the concentration was. Well, you do a similar thing, making standard curves for turbidity. You can make standard curves for oxygen consumption. You can make standard curves for CO2 production. That's what we'll have to pick up next time. We'll spend probably just a few minutes of class on Thursday wrapping up this section, and then I'll let you know that will be the breaking point for exam. Remember, we do have an exam coming up a week from yesterday. Okay? So just leave everything in the incubators for now. You'll be fine until Wednesday.